In the last few segments, we talked about nucleic acids, and we looked at taking the DNA sequence, looking for genes or open reading frames within that DNA sequence, particularly focusing on bacterial systems. And we then evaluated using a codon table how we could take that DNA sequence and predict the sequence of the protein product that that gene would encode. We are going to extend that information out here into the application side of things. And in this video, what we're going to do is focus on some of the main ways that we can go about taking the genes out of one particular organism and introducing them into another, with a focus on taking the gene of interest out of one bacteria and introducing it into some other bacteria. This process we refer to as heterologous expression, where we are taking a gene of interest out of one type of organism, one species of organism, introducing that gene and expressing it in some other organism we refer to as heterologous expression. Why would we want to heterologously express a gene? Well, there's a variety of different reasons, one of which is that we might have predicted a gene of interest from an organism that is really difficult to handle in the lab. Either it grows really slowly or it originated from a really unique environment and therefore is very difficult and tricky to handle in a lab setting. So in those cases, it would be very difficult to carry out experiments by working directly with that organism of interest. But instead, if we can isolate DNA from that organism of interest and introduce some fragment of that DNA that encodes for a gene or genes of interest into a really easy to handle bacteria, then that could make it much more straightforward to understand the function of that particular gene and that particular protein that that gene encodes. And so one common way of going about doing this is to use E. coli as the recipient of the foreign DNA for these heterologous expression experiments. E. coli is very well genetically characterized. It behaves generally very predictably within a lab setting. And so E. coli is a very common host organism for heterologous expression. So in other words, very commonly we take genes out of some difficult to manage or difficult to manipulate bacteria. We take the gene of interest out of that organism, we introduce it into E. coli, and then we use E. coli as the cell that's producing the protein that is encoded by that gene so that we can study that protein. So the focus of this video and what we're going to address by the end of this video is how do we go about taking a gene out of one bacteria and introducing it into another bacterium such as E. coli. So let's go ahead and bring up the whiteboard to get going on this, because we will need to draw some things out here to illustrate what's going on here. So what we're going to be looking at are some terminologies that we will need to know here for this. The first term that we are going to be using in our discussion is the term vector. The vector is going to be a DNA sequence that we can insert the foreign gene into and then take that entire DNA sequence, that is the vector plus the gene we've inserted into that vector sequence, and introduce that whole thing into our host cell, into our E. coli cell, for example. Usually, so the vector we could think of as the DNA sequence that we use as our carrier of the foreign DNA of interest into a host cell. And in most cases, a common vector is a so-called plasmid. So usually, most commonly when you're working with the bacterial system, the most common type of DNA sequence that we'll use to carry foreign DNA into the cell is referred to as a plasmid. A plasmid, as a type of vector, is a small circular DNA sequence, and we can readily introduce, as we'll talk about in a few minutes, foreign DNA into that so that we can propagate that DNA and introduce it into our host cell, such as E. coli. So a plasmid is going to be a small circular DNA sequence. Into which foreign DNA can be readily introduced and propagated. In other words, the foreign DNA can be inserted into this small circular DNA sequence and then introduced into a cell to replicate that DNA. So it's going to be a small circular DNA sequence into which foreign DNA can be introduced and propagated in a host cell. And that host cell is very commonly some laboratory garden variety strain of E. coli. So an example of a plasmid as a circular DNA, we can represent it as a circle on the screen. You will recognize if you're reading scientific papers and they're referring to a plasmid that generally they name the plasmid by putting a lowercase p as the first letter of the name, and then they fill in whatever they want to name the plasmid. So lowercase p there at the beginning of the name emphasizes that this is a plasmid, and we've named this plasmid kind of generically X, Y, and Z. What you will generally see on this plasmid, the small circular DNA sequence, is a gene that encodes for antibiotic resistance. And we represent genes symbolically as these arrows to indicate the directionality of the gene. So in this arrow, the start codon for the gene would be at this end, the beginning of the arrow. The stop codon is at the end of the arrow here, and this is going to be an antibiotic resistance gene, so I'll abbreviate that as AB for antibiotic, and superscript R for resistance. So commonly, if we are working with plasmids that are going into E. coli, this antibiotic resistance gene, when it is present in the cell, will enable the cell to resist kanamycin, ampicillin, chloramphenicol, or some other readily used, widely available antibiotic. The advantage of that is the presence of this antibiotic resistance gene allows an investigator to specifically select for cells that contain this plasmid. So if you're doing an experiment, you're trying to introduce this plasmid into a particular host cell, such as E. coli, you want to make sure that you have a way of easily telling whether that plasmid got into the cell or not. You don't want to have to just randomly look through a whole bunch of colonies of cells and ask yourself, does this one have it? Does this one have it? Does this one have it? Instead, if there is an antibiotic resistance gene in the plasmid, then what you can do is grow the bacteria on petri dishes that contain that antibiotic, and only those bacteria that have this plasmid that confers resistance to the antibiotic will be able to grow. All the other bacteria will not live, they will die, and they will hence not form colonies. So the antibiotic resistance gene is really important because it's going to allow the experimenter to readily select or readily find bacterial colonies on a petri dish that have this plasmid of interest. So if you're trying to introduce this plasmid into an E. coli cell, you'll be able to readily pick out what E. coli cells have that plasmid, because those are going to be the only ones that grow on a petri dish that contains the antibiotic, because this gene allows the bacteria to resist the antibiotics. So this is what we could refer to as the empty plasmid. I'm calling it the empty plasmid because I haven't put any indication here that we have a foreign gene present in this plasmid. What we will do is, through a series of genetic manipulation steps that I'm going to emphasize there with a couple of arrows to indicate it's multiple steps, is we are going to take that empty plasmid, which we called PXYZ, and we're going to introduce the gene of interest into that. So I'm going to call it PXYZ plus the gene of interest. Once that process has taken place, which we'll go through the steps of in a moment, you'll still have the antibiotic resistance gene, but additionally, you will have elsewhere in that plasmid, what will have been introduced is the gene of interest. And this gene of interest that I've illustrated in green here could be a gene that is taken out of some other cell that is perhaps difficult to handle in the lab. That cell wouldn't grow well in the lab or something like that. So it is taken out of that cell, plugged into this plasmid so that it can ultimately be introduced into E. coli and propagated, and the protein product produced for study or for some commercial application, such as the production of insulin or some other protein that is of commercial value. So we'll introduce that target gene in. And once we have done that, we refer to this combination of the empty plasmid vector plus the target gene as our so-called construct. So the construct is specifically referring to the combination of the gene and the vector. The construct, you could think of the term there, you've constructed this, you've created this. That's why we call it the construct. And the way that this is multiplied is that this construct would be introduced into our host cell, and the host cell could be something like E. coli very commonly. Once that construct is introduced into E. coli, we'll pretend this is our E. coli cell. The E. coli will contain our construct of interest, which will have our antibiotic resistance gene in red so that we can easily pick out E. coli cells that contain this construct. It will also contain our target gene of interest, and we would produce literally millions and millions of this E. coli cell, and hence lots and lots, ideally if all goes well, lots of protein product that corresponds to that gene, the gene in green that we had introduced. And so that can have a variety of practical applications, because that protein product could be useful as a protein therapeutic drug. It could be useful as an enzyme to catalyze a chemical reaction that would otherwise be really difficult to conduct using traditional organic synthesis methods. And that can be used for a variety of other things as well. So what we're going to do in the next video is we are going to dive deeper into this process of how do we go from an empty plasmid that just contains an antibiotic resistance gene and no genes of interest to a construct that contains that gene of interest. What sort of techniques can we use to go about that process? And then how can we introduce that construct into E. coli? So we're going to shed some light on the magic of genetic manipulation here. So we will do that in our next segment as we continue onward here.